Hello students, this picture contains complete information about DNA replication and the major enzymes that are involved in this process. DNA replication takes place in 5' to 3' direction. The major requirements for DNA replication are the presence of a template strand, a short RNA sequence, and a free 3' OH group. The two strands of DNA are synthesized in a different manner. One of the strands is synthesized in continuous manner and is known as the leading strand. The other strand, which is synthesized in discontinuous manner, is known as the lagging strand. The major enzymes that are involved are DNA helicase. It results in unwinding of your DNA molecule, which means it converts double-stranded DNA into a single-stranded form. Then your DNA binding proteins will bind to the single-stranded DNA fragment in order to keep it in a single-stranded form. DNA topoisomerase 2, which is also known as DNA gyrase, it will remove the supercoils. In case of eukaryotes, the different types of DNA polymerases that are present are DNA polymerase alpha, beta, gamma, delta, epsilon. While in case of prokaryotes, there are three different type of DNA polymerases, that is DNA pol 1, 2 and 3. The major enzyme that is involved in replication in prokaryotes is DNA polymerase 3, while in case of eukaryotes it is DNA polymerase alpha, delta and epsilon. Your DNA polymerase 3 performs the replication of both the strands in case of prokaryotes, while in eukaryotes there are different enzymes for both the strands. The replication of the leading strand is performed by DNA polymerase epsilon, while the replication of lagging strand is mediated by DNA pol alpha and delta. DNA primase will synthesize short stretch of RNA sequence which is extended by DNA polymerase by addition of nucleotides. In case of eukaryotes, DNA polymerase alpha will initiate DNA replication of lagging strand which is later on taken up by the DNA polymerase delta. Your DNA polymerase gamma is responsible for replication of mitochondrial DNA, while all other DNA polymerase of eukaryotes are involved in nuclear replication. In addition to this, eukaryotes require a sliding clamp protein in order to hold DNA polymerase to the targeted template strand, and it is performed by pCNA, that is proliferating cell nuclear antigen. It will hold your DNA polymerase to the DNA template. In case of prokaryotes, DNA polymerase 1 has an additional activity, that is 5' to 3' exonuclease activity, because of which it will remove the RNA template from the DNA RNA hybrids, while in case of eukaryotes there is no such DNA polymerase which will have 5' to 3' exonuclease activity, so this function is mediated by RNase H. RNase H will remove the RNA fragments. The gap will then be filled by DNA polymerase, and finally the two DNA fragments are joined with the help of DNA ligase, which joins DNA fragments by the formation of phosphodiester bond. However, there comes another problem which is known as end replication problem, in which the end of the chromosomes shortens with each replication cycle. The enzyme that overcomes this problem is telomerase. It is a reverse transcriptase having an inbuilt RNA sequence. This particular enzyme is highly active in case of cancerous cells, resulting in their immortality. Thank you.